Okay. So the second type of complex permanent tissue is phloem or bast. It is a food conducting tissue. So it transports food materials from the leaves to other parts of the plant body. So we'll again be discussing about that in plant physiology, where we are studying about translocation of organic solutes. That is, food is transported from the site of synthesis, that is the source region is the leaf, to the region of utilization, that is the sink region. It might be the root, it might be the tip of the stem where food is required. So that we call it as that movement, transport of food from the site of synthesis or source region to the region of utilization or sink region. We call that as translocation of organic solutes or transport of food. So we'll be discussing that later on in plant physiology. And that is brought about by the phloem. So it transports food materials from leaves to other parts. That is, the phloem transfers food material from the leaves to other parts of the plant body. So in angiosperms, the phloem is composed of sieve tube elements. That is, the sieve tube is the only living cell without nucleus. In the plants, the only living cell without nucleus is sieve tube. So sieve tube are found in close connection with companion cell. So they also have phloem parenchyma and phloem fibers. So in angiosperm, phloem is composed of sieve tube elements, companion cell, phloem parenchyma, and phloem fibers. Okay, the gymnosperms have albuminous cells and sieve cells. So the gymnosperms are having this albuminous cells and sieve cells. They lack sieve tubes and companion cells. So the gymnosperms are lacking sieve tube and companion cells. So please remember about that. In angiosperms, they have sieve tube and companion cell uh, found in close association with each other. We call them as sister cells. Then there is phloem parenchyma and phloem fibers. In gymnosperms, they have albuminous cells and sieve cells. They do not have sieve tube and companion cells. They also have phloem parenchyma and phloem fibers. Apart from this, sieve cells in gymnosperms. Sieve tube is found in higher plants like angiosperm. Sieve cell is found in lower plants like gymnosperms and pteridophytes. So what are the phloem element? If you look into the longitudinal section of this phloem, so you find the sieve tube is arranged one above the other like a, it's in a continuous tube-like structure. You find the sieve tube and the cross walls, if you notice, they have this sieve plates. Sieve plate is just like the gelde plate that we have. Gelde editivella. So for sieving the chaff from the floor. So you are using this sieve plate. So similar to that, the cross walls of the sieve tube, they are having this sieve plate. They have this numerous pores on them and they interconnect this sieve tube. So the sieve, the cross wall or the plates, they are made up of sieve plate. Okay, so they are continuous, they are arranged one above the other sieve tube, and the sieve tube is found in association with the companion cell. You can notice the companion cell which has nucleus. Sieve tube does not have nucleus, but it is a living cell. So the absence of nucleus in the sieve tube is compensated by the presence of nucleus in the companion cell. So the sieve tube and the companion cell, they are considered to be the sister cells. They are the sister cells. So if you look into this structure, the sieve tube is a long tube-like structures arranged longitudinally and they are associated with the companion cells. The end walls of the sieve tube are perforated to form sieve plates. The end walls of sieve tube are perforated to form the sieve plates. So sieve plate with sieve pores, you can see here. A mature sieve element has a peripheral cytoplasm and a large vacuole but lacks nucleus. So peripheral cytoplasm you can find. So at the periphery, and they lack nucleus. They are enucleate. Sieve tube is enucleate. So remember that the only living plant cell without nucleus is sieve tube. Remember about that. So they have a large vacuole but lacks nucleus. The function of sieve tubes are controlled by the nucleus of the companion cell. So I told you that they are closely associated and they are the sister cells. So one depends upon the other. So the function of sieve tube is controlled by the nucleus of the companion cell. So even here you have this protophlloem and metaphlloem. The first form, primary phloem, consists of a narrow sieve tube. So they are very narrow. So we call that as protophlloem. The later formed phloem elements, they have a bigger sieve tube and we call it as metaphlloem. So the protophlloem and metaphlloem, even in case of phloem, you notice that the first form phloem elements which have a narrow sieve tube, we call it as protophlloem. The later form, the phloem, which have a larger or bigger sieve tube, we call them as metaphlloem. So the function of the sieve tube is conduction of food material from leaves. So they are helping in the translocation of food material or transport of food material from leaves. That is the function of sieve tube. So this is the diagram that you are expected to draw. The textbook diagram, you can see towards the right side, the slide of it. So this is the sieve pore. So you can notice this inside the cytoplasm is there at the periphery and they don't have nucleus. That is the sieve tube where I'm showing the cursor. You can notice that is the sieve tube. So they have a sieve plate. End walls are perforated with sieve plates. So you can see the sieve pores in one section. The other one is the sieve plate. You can notice the sieve plate here. And associated with the sieve tube is the companion cell with nucleus. And the companion cell and the sieve tube, they have this plasmodesmatal connection. The functions of sieve tube is controlled by the nucleus of the companion cell. You also find the sieve tube is associated with phloem parenchyma. So these are the components of phloem. For 2 marks, they might ask you draw the longitudinal section of phloem. Uh, so LS of phloem, you are expected to draw this diagram, the simple diagram. Okay, so this is what you have to remember about this diagram. Is they frequently ask for 2 mark question. So the companion cells, let us understand about them. I always say sieve tube and companion cells like sister cells. Uh, Maybe not politics, just for fun. Jayalalitha and Shashikala, that is how their association is. One cannot live without the other. So it is almost the companion cells and sieve tube are of the same nature. They are sister cells and they cannot live or exist without one another. So the companion cells are specialized parenchymatous cells. They are associated with sieve tube elements. The companion cells are specialized parenchymatous cells which are associated with sieve tube elements. The sieve tube elements and companion cells, they are connected by pit fields. So you can notice in the left slide, they have pit fields where they have this protoplasmic connections are connected by pit fields present between their common longitudinal walls. These are the common longitudinal walls. They have pit fields. So the companion cell is connected to the sieve tube by pit fields present on their longitudinal walls. The function of companion cell is to maintain the pressure gradient in sieve tube. The major function of companion cell is to maintain the pressure gradient, that is difference of pressure. Pressure gradient is nothing but difference of pressure is maintained in the sieve tube by the companion cells. The phloem parenchyma, it is found, the parenchyma associated with phloem, we call it as phloem parenchyma. They are thin walled cells and the primary layer of cell wall is made up of cellulose. So all those features are same. So the phloem parenchyma is made up of elongated tapering uh, cylindrical cells. They have dense cytoplasm and nucleus. The cell wall is composed of cellulose and it has pits through which plasmodesmatal connections exist between the cells. So there is a plasmodesmatal connections that is existing between the phloem parenchyma cells. Phloem parenchyma is absent in most of the monocots. This point you have to remember. In most of the monocotyledonous plants, phloem parenchyma is absent. Okay, most of them. That means a few of them have phloem parenchyma. The function of phloem parenchyma is it stores the food material and other substances like resins, latex, and mucilage. So the function of the phloem parenchyma is it stores the food materials and other substances like resins, latex, and mucilage. They are stored in this phloem parenchyma. So phloem parenchyma is made up of an elongated, tapering, cylindrical cells. So you can see this phloem parenchyma, which have dense cytoplasm and nucleus. The cell wall is composed of cellulose, the primary layer of cell wall of phloem parenchyma. It is composed of cellulose, and it has pits through which plasmodesmatal connections exist between the cell. So the phloem parenchyma has pits through which plasmodesmatal connections are existing between the cells. So phloem parenchyma is absent in most of the monocotyledonous plants. And the function of phloem parenchyma is it stores the food material and other substances like resins, latex, and mucilage is stored in this phloem parenchyma. That is the role of phloem parenchyma. Now, the sclerenchyma associated with phloem, so we call it as phloem fibers, or we also call it as bast fibers. One of the good examples for bast fibers is this jute is an example for bast fibers, or twine dara, what we call it as. So from which we make the gunny bags or gonichila. So they are the very good examples for bast fibers. So they are the sclerenchymatous fibers which are associated with phloem. We also call it as bast fibers. Okay, in the slide you can see the fibers. They are elongated with tapering end walls, and they have this lumen which might be septate or aseptate. So you know about that. Then the sieve element here you can notice, and the sieve areas are also noticed here on the sieve element and companion cell. That is another diagram. So the phloem fibers, or it is also called as bast fibers, they are made up of sclerenchymatous cells. So they are absent in primary phloem. So the sclerenchymatous cells are absent in the primary phloem. So these sclerenchymatous fibers, they are elongated. So if you look into the fibers, they are elongated, unbranched, and they have pointed needle-like apex or apices. Apices is bahuvachana, plural form. So they have pointed needle-like apices. So the bast fiber or phloem fiber are not found associated with primary phloem. They are associated with the secondary phloem, and the phloem fibers are elongated, unbranched, and have a pointed needle-like apices. So this cell wall is quite thick because of lignification. The secondary layer of cell wall is lignified, that you can notice in the fibers. At maturity, these fibers lose protoplasm and are dead cells. So the bast fibers or this phloem fibers at maturity, they lose protoplasm and they are dead cells. So the phloem fibers of jute, the I told you about the twine dara from which we make the jute fibers. So the phloem, they are phloem fibers. Phloem fibers of jute, flax, and hemp are commercially used. So the phloem fibers of jute, flax, and hemp are commercially used. The function of phloem fibers, they provide mechanical support and protection to soft tissues. The function of the phloem fibers is they provide mechanical support and protection to soft tissues. So that you can notice in case of the phloem fibers, they provide mechanical support and protection to soft tissues. So overall, the flow chart for understanding the plant tissues, we have this flow chart here. So plant tissues are made up of meristematic and permanent tissues. So meristematic tissues, they are of different types. Apical meristem, intercalary based on their position. The apices of root and stem, they have this apical meristem. So root apical meristem and shoot apical meristem. Intercalary meristem, they are found in between mature tissues. Secondary or lateral meristem, they are found within the mature tissues and they bring about secondary growth. Okay, the permanent tissues are of two types: simple permanent tissue, complex permanent tissue. So simple permanent tissue, they are homogeneous tissue and do not have the ability to undergo cell division. They are of three types: parenchyma tissue, they are the living tissues; collenchyma is living mechanical tissue; sclerenchyma, which is the dead mechanical tissue. So the permanent tissue is also classified as complex permanent tissue, where they are heterogeneous in nature and they are of two types. The complex permanent tissue is of two types: xylem and phloem. So xylem will be having the tracheids in case of pteridophytes and gymnosperms. They have trachea or vessels in case of angiosperms. They also have xylem parenchyma and xylem fibers. All of these elements together, they are functioning as a unit for conduction of water. Similarly, in phloem, the phloem of angiosperms, they consist of a sieve tube associated with companion cell. Then they have this phloem fibers, phloem parenchyma in the xylem. Similarly, the complex permanent tissue in the case of the gymnosperms, they are going to have sieve cells. Then they also have this phloem fibers and the uh, phloem parenchyma. The only living component in case of the phloem, the only dead component in case of phloem tissue is the phloem fibers. The only living component in xylem is the xylem parenchyma. So this might also be asked, so you should be aware about it. So this is the flow chart for the types of plant tissues and their classification of plant tissues, subclassification and classification of plant tissues. So that is about the phloem or the bash that we have completed. The next thing that we would be studying is the tissue system that the plants have. Okay, so that is the thing that we would be studying about. And if you look into the background uh, picture, it is nothing but the monocot stem where they have the scattered vascular bundles. And if you look into the vascular bundles, which are all scattered, and the larger eye part of it is nothing but the metaxylem. Smaller ones are the protoxylem. So if you look into this uh, scattered vascular bundles, they almost look like a skull or a joker. So that is very easily, it is identifiable in case of monocot stem. They have the scattered vascular bundles with a skull-shaped vascular bundle or joker-faced vascular bundle. So it's easy to uh, identify them. So the tissue system, we'll try to understand. After meristematic tissue, they mature to form the epidermal tissue system, so the ground tissue system and vascular tissue system. So based on the structure and location, the tissue systems are of three types. So the epidermal tissue system, based on their structure and location. So epidermal tissue system, they are towards the periphery, the ground or fundamental tissue system, and the vascular or conducting tissue system. So there are three types of tissue system based on the structure and location. The tissue systems are of three types: epidermal tissue system, ground or fundamental tissue systems, vascular or conducting tissue systems. So the epidermal tissue system, it forms the outermost covering of the whole plant body. So, okay, so the epidermis tissue system, it forms the outermost covering of the entire plant body. It comprises of epidermal cells, stomata, and epidermal appendages like trichomes in case of stem and in the root hairs in case of roots. So the epidermal tissue system is found out composed throughout the entire plant body. Even the leaves have lower epidermis, upper epidermis, stem has epidermis, roots has epidermis. So the outermost covering part of the whole plant body is this epidermal tissue system. The epidermal tissue system is consists of epidermal cells, stomata, Then epidermal appendages, like it can be trichomes, which are multicellular, and root hairs, which are unicellular. Okay, so these are the components of epidermal tissue. That is epidermal cells, stomata, epidermal appendages like trichomes and root hairs or hairs. So epidermis, it is the outermost layer of the primary plant body. Primary plant body. Epidermis is usually single layered, okay, and they are almost barrel-shaped cells. And you find the a layer of cuticle above this epidermis, a waxy layer of cuticle is found above the epidermis. In case of leaves, it would be thicker. They have a waxy layer of um, that is cuticle is found. Cuticle is absent in case of roots. The epidermis of root, they do not have cuticle. So epidermis is usually single layered. It is made up of elongated, compactly arranged parenchymatous cells with small amount of cytoplasm lining the cell wall and a large vacuole. They have small amount of cytoplasm lining the cell wall and they have a large vacuole. What you notice, this empty space is nothing but the large vacuole. The outside of the epidermis is often covered with a waxy thick layer, that is a cuticle, which prevents the loss of water. So cuticle is absent in roots. So whenever you are describing about the epidermis, which is present in the epidermal tissue system, it is the outermost layer of the primary plant body. Epidermis is usually single layered. It is made up of elongated, compactly arranged parenchymatous cells with uh, small amount of cytoplasm lining the cell wall, and they have a large vacuole. The outside of the epidermis is covered by a waxy, thick layer, that is the cuticle. It prevents the loss of water. So cuticle is absent in the epidermis of roots. So stomata, they are present in the epidermis of leaves. They regulate transpiration and gaseous exchange. So stomata is present in the epidermis of leaves, especially in dicot leaf. The lower stomata has lower epidermis has stomata, so we call it as hypostomatic. And in case of monocot leaf, both the upper epidermis and lower epidermis, they have stomata, so we call it as amphistomatic. So the distribution of stomata in dicot leaf is towards the lower epidermis. So we call it as hypostomatic. And the distribution of stomata in case of monocot leaf is both in the upper epidermis and the lower epidermis. So since they are equally distributed in upper and lower epidermis, we call it as amphistomatic. So they are present. Where are the stomata located? They are present in the epidermis of leaves. What is its function? It brings about transpiration and gaseous exchange. The exchange of gases, carbon dioxide, oxygen, everything happens there. What is transpiration? The process of loss of water in the form of water droplets, the process of loss of water in the form of water vapor through the stomata or lenticels or cuticle, we call it as transpiration. The process of loss of water in the form of water vapor through the stomata or lenticel or the cuticle, we call it as transpiration. If it is lost as water droplet, we call it as guttation. If it is lost as water vapor, we call it as transpiration. And there is gaseous exchange, that is entry of carbon dioxide required for photosynthesis, also takes place through stomata. There might also be diffusion of respiratory gases that might be happening. So through the stomata. A stomata is made up of two bean-shaped cell or kidney-shaped cells. In case of dicots, the guard cells are bean-shaped or kidney-shaped cells. In monocots, the guard cells are dumbbell-shaped. You can see the dumbbell-shaped uh, guard cells in case of the monocotyledonous plants. So the guard cells are also richly filled with chloroplasts. They also undergo photosynthesis. Each and every cell of the leaf is involved in photosynthesis because leaves are the region where food is synthesized, isn't it? Leaves are the kitchen of the plant. You should remember. So in monocot plants, the guard cells are dumbbell shaped, and in dicot plants, the guard cells are kidney shaped or they are bean shaped. So remember about that. So here the diagram shows the open stoma and closed stoma. So opening and closure of stoma, you can notice majority of plants during the daytime, the stoma is open, and during nighttime, the stoma is closed. There are certain plants like crassulation, acid metabolic plants, which are like call center employees, I call them as, because they have their stoma open during nighttime and closed during daytime. So the stoma is open during the night time and they are closed during the daytime. This is exceptions. CAM plants, we call them as crassulacean acid metabolic plants. They exhibit this type of opening and closure of stoma. Opening of stoma is during nighttime and closure of stoma is in the daytime. Majority of plants, the opening of stoma is in the daytime, and during nighttime, the stoma is closed. So now if you look into the stomata structure, you have to uh, practice this diagrams for 2 marks. They might give it as, see, in case of the bean-shaped guard cells, which you notice in dicot plants, they are kidney-shaped or bean-shaped guard cells. So they are having an outermost thin layer of wall. Inner walls or the inner walls are thick in case of stoma. Okay, and they have large number of chloroplasts in the guard cells. Guard cells are also having chloroplasts. So stoma is the opening which is having guarded by these guard cells. In case of dicots, it is kidney shaped or bean shaped guard cells. In case of monocot, it is dumbbell shaped guard cells. So the opening is called as stoma. The guard cells are also living cells which are filled with chloroplasts. Surrounding the guard cells are the subsidiary cells. Surrounding the subsidiary cells are the epidermal cells. Even in your practical, you are going to take the leaf peel of the epidermal peel, you are going to take it and you are going to stain and observe the stoma. So that micro preparation is also there. So the epidermal peel, you'll be doing it in case of your practicals. So remember about that.
So,
Uh, yes.
so for the guard cells, the kidney-shaped one and the dumbbell-shaped one, is one more advantageous than the other one?
Maybe one is evolutionary point of view, they might have evolved, but I've not come across any importance in the shapes of it. Okay, majority of the monocotyledons. Now, if you ask me why the monocotyledon plants are trimerous, so I'm just saying facts. They are having in threes or in multiples of three. So that is trimerous nature. So most of the monocot flowers, they are pollinated by wind. They are not pollinated by insects. So they are colorless. Why they are so? One is genetic makeup, and another it might have its role, but I have not come across any information as to how it is advantageous to have the kidney shaped or the dumbbell shaped uh, guard cells. Okay, so as far as it, why monocot should have this kidney, the guard shape, the dumbbell shaped guard cells, genetic makeup. One is that, but do they have any specific function? Is it advantageous, more advantageous than kidney shaped? That sort of information I've not come across. It's a good question. Maybe if there is no such information, people like you should work in the future and do research on such things. For that to understand what is the importance or how they might have evolved, we should study phylogeny, that is evolution. So during the process of evolution, how they got separated into kidney shaped guard cells and dumbbell shaped guard cells, that you can notice. So only thing is, whatever the shape of the guard cells, the inner walls, so the inner walls are thick, the outer walls are thin. So to open the stoma, considerable amount of energy is required. I always say it as like a Lord Hanuman opening his chest to reveal uh, the Rama and Sita. So similar to that it is. For them, the thick wall is there. The inner thick walls are there. So whenever the stoma has to be kept open, so they require considerable amount of energy to keep the stoma open. and whenever they are the cells, the guard cells become flaccid, they becomes flaccid, they are going to collapse, they are going to close. Okay, so even in guard cells, inner walls they are thicker when compared to the outer walls of the dumbbell shaped guard cells. One is it is genetic makeup, another is their evolutionary point of view, which is important. I've not come across that information. Okay, you can try to search it in the website. If you get that, you can share it with us also. Or if there is no such information, people, the future generation, they have to work on that aspect of it. It's a good question. If there is no information, research can be done on that topic also.
Okay,
Okay.
sir. Thank you.
Yeah. See, you can also Google out and try to find out and share the information with us if you get that. Like, what is the advantage of a guard cell being bean shaped or dumbbell shaped, which is more advantageous, or what is the evolutionary point of view, why they should be having a kidney shaped guard cell, why they should be having a dumbbell shaped guard cell. Okay, so I have not come across this question. It's a good question. Okay, if there are no answers, research itself can be done on that topic. Fine.
Yes, sir.
So
I'll let you know.
the outer walls of guard cells. See the outer walls of the guard cells that is away from the stoma. This opening we call it as stoma or stomatal pore or stomatal aperture. So the outer walls are thin and inner walls are towards the stomatal pore. They are thick. I was just talking about that. So even when you're drawing diagram, you can thicken the inner walls. Okay. The guard cells, they possess chloroplast and they regulate the opening and closing of stoma. So whatever the shape of the guard cells, they are involved in the opening and closing of stomata. Okay. Sometimes some epidermal cells near the guard cells, they become specialized in shape and size. They are called subsidiary cells. See, there is one more types of stomata based on the subsidiary cell arrangement. Degree level, you'll be studying that. I'm not giving you those information. See, based on the subsidiary cells arrangement, there might be tetrahedral stomata. There are different types of stomata types on that. Okay, so we are not discussing the types of stomata based on the arrangement of subsidiary cells. So, but you can notice subsidiary cells. So for your study, that is not required. So I'm not going into that information. So some of the epidermal cells near the guard cells become specialized in shape and size. They are called the subsidiary cells. So the stomatal aperture, guard cells, and the surrounding subsidiary cells, they are together called as stomatal complex, or you can call them as stomatal apparatus. See the stomatal aperture, you can also just call it as stoma, or you can also call it as stomatal pore. Okay, the guard cells and the subsidiary cells together they constitute the stomatal apparatus, or you can also call it a stomatal complex. Okay, so that is what you can notice on the epidermal cells. So we discussed what is the type of epidermis that we have. Then we are discussing about stomata. The outer walls of the guard cells are thin when compared to the inner walls towards the stoma. So away from the stoma is the outer walls. They are thin. Towards the stoma, they are thick. Okay. The guard cells possess chloroplasts and they regulate the opening and closing of stoma. Sometimes some epidermal cells near the guard cells, they become specialized in shape and size. So some epidermal cells near the guard cells, they become specialized in shape and size, and they are called the subsidiary cells. Based on the arrangement of subsidiary cells, type of arrangement, there are different types of stomata, so which I'm not discussing at present. If you take botany at degree level, you'll be studying that. Okay, so the stomatal aperture, the guard cells, the subsidiary cells together, what do we call them as? We call them as stomatal complex or stomatal apparatus. So the stomatal aperture or stoma, the guard cells and subsidiary cells together we call them as stomatal complex, or we also call them as stomatal apparatus. Okay, so that would be sufficient for today's discussion in the class. We'll continue with the other types of tissues, epidermal tissue in the next class. You have any doubts or queries, you can get it clarified.